This is Dr. Martin's supplemental lecture on viruses, part 3. I'm going to talk a little bit about one particular virus that's called the HIV virus, and this is a virus that can cause AIDS. I'm going to use figure 1624 in your book to talk about this virus. This is actually an RNA virus, and we're going to follow how it reproduces. Couple of things about this virus first. Obviously, number one, it causes AIDS. Number two, it's an RNA virus. Number three, it infects only T helper cells in the body. And these cells are actually part of the immune system. So they're very specific regarding the type of cell that they infect. Okay, so we're going to follow the infection of a cell. So this is a cell here. I'm going to start right here. So this is infection. And the HIV virus has several strands of RNA, as well as an outer envelope with proteins on it. Also has an enzyme. The surface of the virus recognizes or can attach to certain protein markers on the surface of the T helper cells, and that's how it actually attaches to a T helper cell. So in this first diagram here, you see the virus attaching and then releasing its contents into the T helper cell. So this is the RNA, and attached to the RNA is an enzyme, and this enzyme is called reverse transcriptase. And it's actually the target of a number of drugs that are used now to combat AIDS, to keep the viral levels very low. So the RNA is released into the cytoplasm. The next step is that RNA is converted to DNA by this enzyme. So now we have double-stranded DNA. This is actually HIV DNA. That DNA is transported into the nucleus. And actually, let's follow our steps here. So this is reverse transcription to make HIV DNA. Okay, the third step is called integration. And this is where the viral DNA actually is transported into the nucleus of the T helper cell. So this is the nucleus. And that piece of viral DNA is going to insert itself into the host DNA, into the one of the chromosomes of the T helper cell. So this is viral DNA. The next step is that the virus is now going to dictate, take over some of the machinery to make proteins and to make more copies of its genome. So the next step is transcription and translation, so that we end up with RNA and protein of the virus, so viral RNA and protein. So here we've got RNA pieces as well as proteins that are part of the virus. Those components are going to assemble into the virus particle, and that particle, these are all viral proteins, that are on the surface of the envelope of the virus, and this is called budding. So what happens is that this cell is going to release particles of the HIV virus that can then infect other T helper cells. At some point, the cell might actually be destroyed. There are some people who are not infected by HIV, even if they're exposed to it, because this very first step, these individuals lack a specific protein on their T helper cells, and that means that the virus cannot attach because there's no docking site on their cells. So they're essentially, they're not infected. They won't become infected even though they're exposed, could be exposed to HIV. Okay, so this was 16-4, and I think that is the end of this supplemental lecture.